Today we're going to be hearing from Michalis. His talk will be about 20 minutes, and then there'll be time for questions and discussion afterwards. Just please keep yourself muted during the talk, but feel free to post questions in the chat. Yeah, as maybe I'd mentioned a minute ago, we're recording the session and you can watch it again afterwards on YouTube. And yeah, I think that's all the housekeeping, and everyone's quite familiar with that by now. So yeah, a little bit about Michalis. Michalis is a PhD candidate at the Institute of Molecular Biology and Biotechnology in Greece. And today he's going to be talking about incorporating dendrites into spiky neural networks. He had a preprint on this recently, which I'll post in the chat. And if you want to learn more about it, you should go and read that as well. And yeah, I think in the community, the DAMS lab in particular, we're really excited about the idea of being able to have SNNs with dendrites. And also, I guess from an experimental point of view, excited about the possibilities which being able to model dendritic computations better might open up. So yeah, with that, I'll hand you over to Michalis.
Thank you very much, Marcus, for the introduction. And also thank you for inviting me in this amazing seminar series. I have been a fan since its beginning, so I didn't expect I would be invited to present my work here. So without losing time, today, for the next 20 minutes, I'm going to talk a lot about dendrites. I will start with presenting you why we think that dendrites can be conceptually useful and important. Then I'm going to present you some strategies on how to develop the SNNs that can reproduce accurately some key dendritic properties. And finally, I'm going to present you Dendrify, what it is and what we can actually do with that. First of all, I would like to start with this typical image of a cortical pyramidal neuron. You can easily distinguish its three main parts, being the soma, that is the main part of the cell that contains the genetic information, the axon that transmits information to other neurons, and the dendrites that actually receive information from other neurons. And for many decades after the discovery of pyramidal neurons, people had no idea what dendrites do. They simply thought that they are just passive propagators of synaptic input. So Michalis and Pitt in 1943 came up with a point-neuron idea, or the perceptron, as you may know it, that actually what it does, it doesn't care much about dendrites, so it simply adds linearly synaptic inputs and then passes into a nonlinear activation function. This simple model accurately mimics what happens at the axonal initial segment of the soma. And it proves quite useful so far, and until now it's a foundation of modern ANS and deep learning architectures. And it was so useful and that actually even the spiking neural networks are inspired by that. And although artificial neurons and spiking neurons have many similarities, I'm here to argue that they have one major, actually they have many differences. I'm here to argue that they have one main similarity. And according to newer data, they're not so accurate representations of how real neurons actually compute information. And whenever I hear about neural networks that don't care much about dendritic computation, this image comes in mind. I mean, they do the job, they do amazing stuff, but they are not how they were supposed to be or how they were designed by nature. So to understand why we believe that, first of all, I would like to note that dendrites, like the soma, they're able to produce action potentials, but unlike the soma, these action potentials come in many different flavors and they operate in multiple time scales. First of all, they can be sodium spikes that they can last for a couple of milliseconds. They can be calcium spikes that they last for tens of milliseconds. And they can even be an MDA spikes that they can last for up to a second, let's say. Moreover, the direction of these spikes is not like the axon that is usually unidirectional. They can be produced at the dendritic location and then travel to the soma and to the axon, or they can be generated at the perisomatic domain and then travel backwards to the dendrites. And as such, they act as feedback signals of neuronal activity. Moreover, the situation is even more complex because we now know that dendrites can increase greatly the heterogeneity of neuronal computations. Their passive properties, like the morphology, their cable properties, in conjunction with the voltage-sensitive ion channels they have, they can generate a plethora of different activation functions, all of which can coexist in the same neuron. Moreover, apart from the neuronal properties, also synaptic properties play a major role in the transformation function, the transfer functions of neurons, since synaptic weights or the distance between inputs can greatly impact the linearity of the response. Synaptic allocation can greatly impact as well how neurons compute information. For example, when you cluster many synaptic inputs into a single branch, the probability of triggering some dendritic nonlinearity is much higher. Whereas if you spread these synaptic inputs in different branches, neurons tend to respond in a linear manner. Another key feature of real neurons is that usually they receive multiple streams of information that they are also spatially segregated along the dendritic trees. To simplify this image a bit, let's have this simple example here. So the whole distal domain of the neuron shown here in the cortex is supposed to receive contextual information from higher order areas, and whereas the proximal domain or the basal, the dendrites, are supposed to receive the sensory information. And what is the functional implications of this? So when we have a single pathway being activated at a distal branch, we know that due to intense somatodendritic attenuation, the effect at the soma might be quite weak. Whereas when you have, I'm sorry for that, when you have inputs being more proximal to the soma, they are less prone to being attenuated. So you have moderate somatic activation. However, when you combine distal and proximal inputs, you have the generation of some intense dendritic nonlinearities, some long-lasting events, usually they are calcium or NMDA spikes, which result in the neuron firing in bursting mode. Moreover, apart from having an effect on the output of the neuron, the segregation of pathways can have a meaningful impact at the synaptic plasticity. For example, when you activate locally some pathways, you can have the local generation of dendritic spikes that can trigger synaptic plasticity regardless of somatic activity. Something similar happens also at the proximal part of the dendrites, but this is more Hebbian-like because usually they are more coupled to the soma. Whereas when you activate both pathways, both distal and proximal pathways, you have global and heterosynaptic LTP due to strong and long-lasting plateau potentials. So I just mentioned some basic, well-known functions of dendrites, and the next question is, how do all these things we mentioned impact network-level functions? To answer this question, we had to come up with a new model of dendrites, because traditionally, when we studied dendrites, we used to work with some complex and biophysically detailed models. Although they are super realistic and highly accurate and they allow for low-level experiments, they're not so efficient. They can be complex and they are impractical when we want to use them in neural networks. On the other side, reduced integrative firing models can be lightweight, they can be mathematically tractable, they allow for building large networks. However, they are less accurate, unrealistic, and they don't have dendrites. So we thought, why don't we try to combine best of both worlds? And that's how the idea behind Dendrify emerged. And to showcase how we see things, so this is a point neuron that we used to create when we wanted to model a whole pyramidal neuron. Okay. So we thought, why don't we just use a couple more points that they can represent whole dendritic neighborhoods of the neuron? So, for example, this pyramidal neuron can be broken into 3 territories: one distal territory, one proximal, and the one that represents the soma and the basal dendrites. By the way, this segregation is arbitrary, depending on the experiment or the research question. You can break it in more or less. It's just for this example, so bear with me. This is what we call traditionally as the somatic layer, of point neurons, which acted also as the output that is passed maybe to other layers. So we suggest that we can extend the point neurons by adding some extra nodes, forming the proximal and the distal dendrites. All together, they can create 2 extra dendritic layers. So instead of having a single layer representing 1 neuron, now you can have 3 different layers, all of which, where the compartments, of a unit extend in all these layers. And why is this important? What are the key features of this architecture? First of all, we have somatodendritic decoupling. In real neurons, we know that synaptic integration and plasticity do not operate on the soma, but they are semi-independent of that. So this architecture can achieve this. And also, another thing we can do is to experiment with coupling strength. We can have this as a learnable parameter and have, let's say, a heterogeneous somatodendritic decoupling in a neural network. Another thing that is really important is that each node can have a unique set of active mechanisms. You can have sodium or calcium or NMDA spikes, all of which contribute differentially to the global function of the neuron. Another thing is that because these dendritic nodes are usually they're thinner and they are smaller, they are more excitable. Thus, they have a lower threshold for local spiking and plasticity. Another implication is that we can have a more realistic interaction of multiple input streams. This architecture allows us to have a single input stream that mimics what actually happens in real neurons. If you remember that one pathway goes to the distal part, another pathway is going to the to the proximal part, and when they are in isolation or activated together, we have very different effect at the neuronal output. And lastly, you can have actual backpropagation of information happening within a single neuronal unit, since after the somatic activation, we can have the dendritic nodes also to fire some dendritic spikes. Thus, the information can backpropagate from the somatic layer to dendritic layers. And having explained a bit the theory behind Dendrify, let's see it in action. Let's say we want to build a 3-compartment model like this one. We have a somatic compartment, we have a single compartment for the apical dendrite, and a single for the basal. This is just for illustration purpose. It's still like the 3 dots I showed earlier. The way we describe this with Dendrify is as we see it here. This is the whole code needed to reproduce the model we show here. Each compartment can be represented by a single line of code. Actually, quite is it's descriptive code. You can also specify how the various compartments are connected together and the strength of connection, and some global neuronal properties. And then you can use this neural model, pass it into Brian, and run simulations. Then, like, how would you normally use Brian? We chose Brian because it's a super friendly neuronal simulator. It has really good support. It was just a natural choice for us. And if you work with it, you will figure out that it actually makes modeling much simpler. So let's see some more complicated examples. Let's say here we want to build a 4-compartment model where there are 2 different streams of information. We have some synapses going to the distal, some synapses going to the proximal part, and all dendrites can be active. They can have dendritic spikes. Again, we have this approach we discussed earlier. Everything can be triggered with single lines of code. For example, if you focus on the proximal dendrite, we can activate AMPA synapses and MDA synapses, and we can activate sodium channels with just little code. It's quite straightforward. And what actually Dendrify creates for you in the background is all the equations, the parameters of the models, and many things that they don't show here, like custom events that work for Brian and a lot more. In the past, you had to do this manually, by the way. And let's see this in action. Let's say we inject some current. Oops. Let's say we inject some current on the distal part. It's 5 milliseconds long, and the intensity is barely strong enough to trigger dendritic spiking. You can see how dendritic spikes are locally generated. These are not integrated and Fourier type of responses. I will show you later how it works. And you can see the dendritic attenuation. The somatic compartment barely feels the generation of dendritic spikes in the distal part. You can see something similar happening in the input when the input is provided to the proximal part. One notable thing here, as I mentioned also earlier, the dendritic properties change a lot the input-output function of the neuron. So here you can see that I can trigger the dendritic spike with much more current than I had to work with in the distal part. And the same for the trunk compartment. And some other notable things I would like to say is that when you take the derivative of the voltage response, the response on the dendrites versus soma, you can see how dendritic spiking initiation location affects the somatic compartment. When I check the dendritic responses at the level of dendrite, we see that they are almost identical. But you can see that the effect on the soma varies a lot based on the initiation location. Also, you can see here when we plot the input-output function of the distal and the proximal part, they are almost identical in terms of the sigmoid function. But you can see because the distal part is much more excitable because it's thinner, it requires only 6 synapses to trigger spiking, whereas the proximal part requires 18. Another notable behavior of this model is you can have realistic backpropagation, as I mentioned earlier. You can see when the soma fires an action potential, what happens is that spontaneously, the dendritic spikes can be formed at the trunk and the proximal dendrite, but you don't see anything in the distal part. And these are the so-called spikelets, which are have also been observed experimentally, and there are just some small events that are not enough to trigger further the dendritic spiking. And what I didn't mention, that all these mechanisms we show here, they do not use the Hodgkin-Huxley formalism. To replicate all these phenomena, we use one event-driven mechanism that works like this. When the threshold of a given dendrite is crossed for dendritic spiking, we have the activation of a sodium current that decays exponentially, and that after a few milliseconds, we have the generation of potassium current that also decays exponentially. And together, they try to mimic how sodium spikes are generated with the Hodgkin-Huxley model. And we also compared it. We also compared how the Hodgkin-Huxley model works versus this approach. And as you can see here, if you tune the parameters, you can see they're almost identical. So we can trust that these sodium spikes, they are very similar to how they should be modeled, but the way we model them is much more efficient and requires much less resources and parameters. Okay, I think the time has passed. In summary, neuronal dendrites have numerous properties with perhaps significant implications in the fields of SNNs. Extending point neuron models with dendritic nodes allows simulating many key dendritic functions, and Dendrify provides an easy access to such models by automating their development in Brian 2. The next part of this presentation is still under construction. Now we have the tools, and we're going to test all the research questions we have on how dendrites actually impact network computations. So you have to wait for that. I'm sorry. And I would like to thank my supervisors, Panayota Poiraz and Spiros Havlis. Also, I would like to thank Dan Goodman for his initial feedback on this whole project, and Marcel Steenbeck that is still being super helpful and provide some really good feedback for our reviewers. And all the lab members that create a beautiful and constructive environment to work with, and especially Kostas Petrosakis and Simone Tasciotti, that they were my beta testers, and Nikos Malakasis, who has given me a lot of constructive feedback. So thank you very much. That was all. If you have any questions, please let me know.
Thanks so much for the talk. That was really great.
Thank you.
Yeah, I'm happy to, if anyone has any questions, just either come on camera and ask, or you can type in the chat and I can read it out. I'm happy to go first then. So I guess something I was wondering about was, have you thought about trying to have networks which you can train with either local rules or with surrogate gradient descent, which have these dendritic compartments?
Yes, we have. I think, I don't know if we can actually implement surrogate learning in Brian. Maybe we have to go to shift to PyTorch for this thing. But of course, let me show you. I can find a slide. Here, the next step we thought would be how would some common plasticity mechanisms work when we have dendritic nodes. For example, we can try STDP, and it could take dendritic spiking as the post events, or we can test the BCM. And we would like to extend all the known plasticity mechanisms that work very well for point neuron to this category of models and see how it works out.
Yeah, that sounds super interesting. That's really cool to be able to do that.
It's already in the plan. Here, Dendrify is just the means of creating this type of models, but you can use whatever learning rules you wish. And actually, now you can use dendritic spiking as the input for these rules. For example, in STDP, as I mentioned earlier, you can have the dendritic spikes, not the somatic spikes. So you have super local rules of learning.
And I guess, sorry, I think maybe Dan wants to jump in. He's turned his camera on, but just one more quick question was, could you imagine, I guess, so for now it makes a lot of sense to start with like essentially like 3 or 4 compartments, right? But could you imagine having like more like entire dendritic trees and things eventually?
Actually, we can do this already with tools like the neuron simulator. I mean, typical biophysical models can have hundreds of thousands of compartments, but increasing the complexity, the number of compartments make these models super computationally costly. So the whole purpose of this project was how to reduce the amount of compartments without losing the dendritic computation we wanted to look into. So yes, of course, we could increase the number of compartments, but the whole point was how to decrease them.
Sure, okay. Thanks.
Thank you.
May I go ahead, Marcus? Or?
Yeah, of course. Sorry.
Yeah, so we had this paper recently about how heterogeneity in neural networks could be valuable, and we were just looking at heterogeneity and time constants. I'm wondering, is there a way to sort of with something like this to sort of define what that space of heterogeneity would look like, or would you need to look at experimental data on lots of different sorts of dendritic trees and think about, I mean, like, is there something that you could do easily and quickly, or is it something you'd have to think hard about how to sort of set up an appropriate heterogeneous space of neurons to see what that does.
We were actually inspired by this paper, your paper on heterogeneity. That's why we did a lot to include heterogeneous mechanisms inside a single model. So we introduced heterogeneity in terms of that we increase the number of possible computations that a single neuron can perform. However, if we would like to perform an experiment similar to yours, I think you use trainable membrane time constants. For example, in this network, we could have trainable coupling strength, coupling conductance. So we can have a whole network where this is a free parameter, and we can just use it to, a trainable parameter. And then an experiment would be to see, okay, if we constrain this in a biological manner, is it going to be better at a given task, let's say image classification or something? So it is something that we want to actively explore, how it affects the network performance. I don't know if I answered fully your question.
Yeah. I think it would be really interesting. I'd love to know the results of that. It's true that Brian at the moment can't do any sort of PyTorch-like gradient descent sort of mechanisms. I wonder if in this case, since you presumably produce a set of equations, which you then pass to Brian, it might be relatively easy to generate PyTorch code from that, which we haven't, I mean, it would be relatively easy to do. We haven't really done it just because for a lot of the Brian models, it's almost not worth the effort because they're quite simple models typically. But in this case, the equations are pretty complicated. So it might well be worth the effort to sort of auto-generate some of those.
Exactly. The reason we started with Brian, because it's super easy to learn. It has a small learning curve and it's super intuitive. It has a good documentation. I mean, we wanted to standardize this approach first in Brian, so because it's a very accessible and easy tool for anyone to use. And one of the future orientations of this project is how, if there is a need for that, of course, if it makes any sense to try to replicate what we did in Brian also in PyTorch. So for now, it's not supported because we still haven't tried training SNNs with surrogate descent. But if we need it, perhaps we can have a version of Dendrify that works with PyTorch. I don't think it would be super hard to translate all this to PyTorch.
Yeah, I think I tend to agree. I think it should be quite doable.
Also, by the way, since we have this, anyone who is familiar with PyTorch can try to translate this. I mean, this is completely open access for the community. So if someone thinks that this is a good foundation to start working, they can just take it up to the, up to the neural model level. Here, we don't use anything Brian related, apart from the units, but we can change this also. And they can just experiment how to translate this to PyTorch. I mean, if it's not us that we do this, perhaps someone that is interested in this, in looking into this mechanism and wants to transfer them into PyTorch, just feel free to do it.
Yeah, I'd be happy to help on doing that as well. I think some of the stuff we've written for Brian would make it easy, but yeah. Cool, thank you.
Thank you very much.